Hey guys, welcome to chapter 24, covering digestive system infections. The digestive system's purpose is to break down and absorb food molecules, as you probably know, and then those food molecules pass into your circulatory or lymphatic systems, where they are carried throughout the body to your various tissues and used for energy. The digestive system, also known as the gastrointestinal tract, starts with the mouth, leads to the throat, then the esophagus, the stomach, the intestines, and out the anus. It is lined with mucous membranes, and in particular, the stomach contains one of the more powerful lines of defenses in your body, which is gastric juices that have a pH that is very low, a pH of 1.5 to 3.5. The digestive system also contains accessory structures, which we will consider in this chapter as well, including your teeth, your tongue, your salivary glands, your liver, your gallbladder, and pancreas. As far as normal microbiota go, most parts of the digestive system are heavily populated with microbes. In fact, your digestive system is the most heavily populated body system out of all of them. There are millions of bacteria per milliliter of saliva found in your mouth. There are around 100 billion bacteria per gram of feces in the large intestines, and in particular, the genera Bacteroidetes, Clostridium, the species E. coli, and Archaea that belong to the group Methanogens are prevalent in the large intestine. Your stomach and your small intestine, compared to the large intestine, are relatively less populated, but still contain abundant communities consisting of lactobacillus, corynebacterium, and yeasts belonging to the genus Candida. In all of these, microbiota play important roles in your digestion. They assist in breaking down large molecules, they synthesize and excrete vitamins that your cells take up and use, and they also outcompete transient, opportunistic pathogens by taking up space, by consuming resources, and eliminating opportunities for pathogens to colonize the digestive tract. There are quite a few diseases of the digestive system that we're going to be covering. We'll be looking at bacterial diseases, and we'll split them into diseases of the mouth as well as diseases of the lower digestive system. We'll also be looking at viral diseases, protozoans, and helminthic infections. And we're going to start by looking at bacterial diseases of the mouth, of which there are two. We're going to talk about dental caries and periodontal disease. Dental caries is the medical term for tooth decay. Tooth decay occurs when your teeth accumulate microbes in a biofilm that we know colloquially to be called plaque. So when you eat something sugary and later you run your tongue over your teeth and you feel that fuzzy feeling, that is a biofilm of microbes that are building up on the surface of the teeth. This happens easily because unlike your mucous membranes of your skin, which have access to the immune system and are constantly sloughing cells, your teeth do not have these defense mechanisms. And so they can easily accumulate a biofilm, and when plaque becomes heavy enough, it can thicken and calcify into what we colloquially know as tartar. And this happens primarily as a result of the carbohydrate sucrose. Sucrose is the technical term for table sugar, and as the microbes in your mouth metabolize sucrose, they transform it into two different things. The first of these is dextran, which is the gummy component of plaque, and the other is lactic acid, as well as other organic acids, which lower the pH around your teeth and contribute to destruction of the enamel layer on top of your teeth that protects them. The primary species of bacterium that is involved in this metabolic process is Streptococcus mutans, which metabolizes a wide variety of carbohydrates and also tolerates the high acidity levels that are generated when the sucrose is metabolized into organic acids. Here we can see in these images Streptococcus mutans. We can see that the Streptococcus shape is exhibited, And down here, we see that Streptococcus mutans has now secreted a large amount of dextrin, that gummy substance that is contributing to the production of a biofilm layer. So whereas dental caries is the destruction and decay of the tooth itself, periodontal disease, or second disease of the mouth, is a catch-all term for conditions related to the degeneration of the tissues and support structures around the teeth. Gingivitis is the infection and inflammation of the gums that affects approximately 50% of US adults and is considered a mild form of periodontal disease. Gingivitis, however, can progress, if chronic, to periodontitis, which is the more severe form of periodontal disease associated with the formation of pockets of pus at first, which can progress to the destruction of the root tips that support your teeth, and then eventually lead to the loss of teeth altogether because their support structures have been degraded. So now we're going to move on to bacterial diseases of the lower digestive system. We're still talking about bacteria, but we're moving past the mouth and progressing down into the stomach and intestines. So first, let's take a look at staphylococcal food poisoning. We talked about staphylococcal food poisoning in this section because it is caused by a bacterium. But it's technically not an infection, it's an intoxication. It's caused by ingesting not Staphylococcus aureus itself, but the toxins that are generated by it. We've seen Staphylococcus aureus before. It's this gram-positive and spherical-shaped cells that are arranged in clusters. It causes a variety of different infections, including the staph skin infection, but it can also lead to food poisoning because Staphylococcus aureus produces 21 different thermostable toxins, meaning toxins that can withstand high levels of heat and not break down. It takes approximately 1 million bacteria per gram of food to generate enough toxin to cause an actual disease state, and typically, staphylococcal food poisoning is mild and self-limiting within 24 to 48 hours. But let's take a look at how this happens. A typical scheme leading up to an incident of staphylococcal food poisoning is that food is cooked, and then after cooking, it's contaminated with staphylococci coming from the hands. Remember that 20% of people are permanent latent carriers of staphylococci in their nasal passages, and up to 60% are transient carriers. And so Staphylococcus aureus is all around us, and it's quite easy to transfer it from, for example, your nose to your hands and then to food. But then if that food is left at room temperature or in a warming station in a restaurant, for example, then this temperature is conducive for the staphylococci to multiply and start producing and spitting out those toxins. The toxins are consumed with food, and even if the food is reheated, the toxins remain because they are thermostable. So once they have been produced and they are in the food, they are there to stay, and they will cause food poisoning when that food is consumed. So the best method of preventing staphylococcal food poisoning is refrigerating food during storage, because this prevents Staphylococcus aureus from growing and multiplying and generating those toxins. In this checkpoint here, I'd like you to explain to me why treating staphylococcal food poisoning with antibiotics would not be an effective solution. Next, we have shigellosis, which is a disease that is also known as bacillary dysentery. It's caused by 4 members of the genus Shigella, which is a gram-negative rod-shaped bacterium. These little guys multiply in the small intestines over a 1 to 3 day incubation period after consuming them in food. Of the 4 species in this genus, Shigella sonnei is the most prevalent one in the United States and is associated with mild symptoms. But Shigella dysenteriae is the more severe one that is commonly found in developing countries. And this one, its severe symptoms can be attributed to the production of a very powerful toxin called the Shiga toxin. The results of an infection with Shigella dysenteriae are fever, cramps, and up to 20 bowel movements per day containing blood and pus from intestinal ulcers generated by this bacterium. There are an estimated 500,000 cases worldwide of shigellosis, and the mortality rate is up to 20%, but that mortality rate is really associated with a lack of hydration treatment. The reason why people can die from shigellosis, and for many of these bacterial infections of the lower digestive system, is a result of not being able to access proper hydration through, for example, intravenous solutions, and that's truly why these diseases are so dangerous. Next, we have salmonellosis. Salmonellosis is an infection that is caused by either Salmonella enterica or Salmonella bongeri, but Salmonella enterica is by far the more common of the two. The way that these organisms infect the digestive tract is they pass through the mucous membranes and into the cardiovascular and lymphatic systems and cause a systemic illness. What you see in this image right here are the Salmonella bacteria performing something called membrane ruffling, which is a process by which they are able to penetrate our cells and gain access into the deeper tissues of the body. The incubation period is 12 to 36 hours, and again, this bacterium is typically transmitted through the fecal-oral route through contaminated food or water. It's associated with fever, cramps, diarrhea, nausea, and this lasts a few days to a week. And it's typically self-limiting. There's an estimated 1 million cases and 380 deaths annually in the United States, so a very low mortality rate. But these statistics, at least for cases, if not for deaths, are probably underreported because, again, it's self-limiting and it typically doesn't require medical attention or hospitalization. As far as how to prevent and avoid salmonellosis, making sure to cook foods thoroughly, particularly poultry, because chickens and turkeys are especially susceptible to infections, as well as their eggs. There's an estimated 1 in 20,000 eggs in the United States that is contaminated with the Salmonella pathogen, but cooking everything thoroughly effectively kills the pathogen. Next, we have typhoid fever. Typhoid fever is also caused by Salmonella enterica, but it's caused by a specific serovar, the serovar typhi, and therefore the name of this organism is often just abbreviated S. typhi, Salmonella typhi. It is spread also through the fecal-oral route, but it is now rare in the United States due to sewage treatment. The thing that the species is able to do is multiply in leukocytes that perform phagocytosis. So the leukocytes, they engulf the Salmonella typhi cells, and then instead of being able to destroy them, the pathogens multiply inside of these leukocytes. The leukocytes are circulating in the blood and lymphatic system, and in this way, the organisms are able to sort of hitch a ride in the leukocytes to other parts of the body and spread to different organ systems. After a 2 to 3 week incubation period, the symptoms include headache, fever, and a later onset of diarrhea. Mortality rate is 10% if untreated, but it is very treatable now with antibiotics. There's also a vaccine available for preventing it, but the vaccine only lasts 2 to 5 years before it has to be repeated. Recovered people have lifelong immunity, but 1 to 3% end up being chronic asymptomatic carriers, meaning that they carry this pathogen in their body and exhibit no signs of disease, but are still contagious and able to transmit it to other people. And it is from this capability of the pathogen that the story of Typhoid Mary emerges, which you can see here, an old newspaper article about it. You may have heard of Typhoid Mary before as the story of Mary Mallon, who was one of these asymptomatic carriers. She worked as a personal cook, and she spread through her work this pathogen to 51 people and ended up killing 3 of them as a result. It was a major problem because at the time when she was active, there was a very poor understanding of infectious disease, particularly among lay people like herself. And so she was quarantined once, and then she was let out on the condition that she stopped working as a cook. But she refused to do that and continued to work as a cook and infect people because she didn't believe that she had this disease. And therefore, she had to be forcibly quarantined for the rest of her life because she simply would not stop behaving in manners that would infect and in some cases kill other people. Next up, we have cholera, caused by the species Vibrio cholerae, which is a gram-negative curved rod that has one flagellum by which it is motile. These guys colonize the small intestines and release an exotoxin that has a very specific effect on the cells of the intestines. It causes water and electrolytes to be expelled out of the cells of the intestines, and this results in massive loss of fluid through profuse diarrhea, sometimes called rice water diarrhea. Up to 5 gallons of bodily fluid can be lost in one day through the action of this toxin, which leads to an increase in the viscosity or the thickness of the blood. The blood can become syrupy and less fluid. This can also cause a dangerous decrease in blood pressure known as shock, and then lead to death. This is a very dangerous disease if untreated because of the dehydration factor. Untreated mortality rate is up to 50%, but when treated with rehydration therapy, the mortality rate is only 1%, so it's highly treatable. But it's important to have access to medical services and medical technology that can provide you with intravenous fluids to treat this disease. Next, we have a checkpoint. So the cholera pathogen can be found in coastal waters in Southeast Asia, among other areas of the world. But here, it grows on the surfaces of crustaceans, so it's constantly present in this region. How would we classify the pattern of incidents in this region? Would it be sporadic, endemic, epidemic, or pandemic? Next up, we have E. coli gastroenteritis. So gastroenteritis caused by one of a variety of strains of E. coli. We know E. coli as being a normal, mutualistic, beneficial member of our gut microbiome. Typically, it's harmless, but horizontal gene transfer from other bacteria or lysogeny from a temperate bacteriophage can cause certain strains of E. coli to become virulent, even though they are normally non-infectious. E. coli gastroenteritis is the most common cause of traveler's diarrhea, and there are 6 varieties, 4 of which that we will talk about here. The first is enterotoxigenic E. coli gastroenteritis. These strains of E. coli produce a thermostable toxin that is similar to cholera toxin, but cases of this disease, unlike cholera, are typically mild. Enteropathogenic E. coli, their mechanism of virulence is to destroy the microvilli, or the tiny projections that are similar to cilia that are present on the surface of the intestinal epithelial cells, and they sit there and secrete proteins that lead to diarrhea. Enteroinvasive E. coli will actually invade the intestinal epithelial cells, penetrate them, and multiply inside, causing a systemic illness. And lastly, and most severely, enterohemorrhagic E. coli. These guys release a Shiga-like toxin called verotoxin, which is now known to have been horizontally transferred from the Shigella bacteria. So E. coli sort of ripped off this Shiga-like toxin from Shigella and therefore are able to cause diseases that are similar to dysentery. The toxin in this case is often called verotoxin, and the effect that it has is it can, but does not always cause hemorrhaging in the colon or the kidney. And the one thing that is dangerous about enterohemorrhagic E. coli is that it does have a reservoir that is near to us in domestic cattle. An estimated 30% of domesticated cattle are carriers of enterohemorrhagic E. coli, also known as strain O157H7. In 1999 and in years following, there was some research done that showed that the diet that the cattle are fed has an impact on the ability of their stomach to harbor these bacteria. The cattle are unaffected by enterohemorrhagic E. coli, but their stomachs can be made more hospitable to these bacteria when they are fed grain as opposed to a natural foraging diet consisting of hay. So this is an interesting fact because cattle in feedlots are typically supplemented with corn or grain, and that is done to fatten the cattle up and get them ready for slaughter. But this research suggests that feeding them in this way actually makes them better able to harbor this pathogen and possibly pass it on to humans as well. So next is campylobacteriosis. This is infection caused by members of bacteria in the genus Campylobacter. But the most common species is Campylobacter jejuni. These bacteria are gram-negative and they are microaerophilic in their oxygen requirements, as well as being spiral-shaped, as you can see here. Associated signs and symptoms include fever, cramping, and bloody diarrhea, and these typically resolve within 1 week. The most common incidence of campylobacteriosis is in children aged 1 to 5, and how it is transmitted and acquired is from reservoirs that include poultry and cattle. Virtually all poultry meat and 60% of milk is contaminated with bacteria in the genus Campylobacter. And this is why milk pasteurization is particularly important to clear those bacteria, as well as cooking poultry meat thoroughly. Based on the oxygen requirements, tell me, in this checkpoint, the pattern of growth that you would expect to see in a thioglycolate tube from Campylobacter jejuni. Next, we have peptic ulcer disease caused by Helicobacter pylori. It is also a microaerophile that is spiral in shape with a flagellum or multiple flagella that it uses to move about its environment. Peptic ulcer disease is interesting because it's not completely well understood. It's known that 2 out of 3 people contain Helicobacter pylori as part of their normal stomach microbiome. But out of those who have it, only 20% of them will develop ulcers of the stomach and duodenum. And the risk factors for what distinguishes someone who develops these ulcers versus someone who does not are not entirely clear. What we do know is the mechanism of action exhibited by Helicobacter pylori, which is where they attach to the epithelial cells of the stomach and release the enzyme urease. Urease we've talked about before in microbial mechanisms of pathogenicity. It is an exoenzyme that will produce ammonia, which is basic in pH. And the effect that this ammonia will have is to neutralize the gastric acid surrounding these bacteria and create a more hospitable microclimate for them so that they can then colonize the mucous membranes of the gastrointestinal tract, disrupt the mucus that protects the stomach lining from acid and digestive enzymes, and when they get rid of that mucus, the acid and digestive enzymes can start to irritate the cells that line the stomach because the mucus that would normally serve as a barrier between that acid and your stomach lining is gone. And so this is the mechanism by which the ulcers are able to appear. It is also known that 50% of Helicobacter pylori strains also produce an exotoxin which can exacerbate the effect of this ulcer production. Next, we have C. difficile associated diarrhea, also sometimes just referred to as C. diff. It is caused by a gram-positive rod called Clostridioides difficile, formerly known as Clostridium difficile, but reclassified recently as Clostridioides as a genus instead of Clostridium. This organism is normally a harmless resident of the intestinal microbiome. Lots of people have this organism in their gut. However, overgrowth can lead to symptoms that range from mild diarrhea to deadly inflammation and perforation of the colon. Risk factors for this overgrowth include broad-spectrum antibiotic therapy, old age, and immunosuppression. It is also a nosocomial infection that can be acquired in the hospital. It's very dangerous and is thought to have caused more deaths than all other types of gastroenteritis combined, and is typically treated with antibiotics or a fecal transplant. Now, the fecal transplant is a more recent advent in the treatment of C. diff. Antibiotics were the standard treatment for a long time, but at some point it became clear that an infection that precipitates as a result of antibiotics may not be best treated with antibiotics. So what a fecal transplant is to take stool samples from healthy individuals and that have been screened for any sort of pathogens and transfer them to individuals who need to have their microbiome remediated or built up again due to weakening through, for example, antibiotic treatment. So as you can see in this article, stool transplants are now part of the standard care for recurrent C. diff infections. And those stool transplants can happen from either direction. They can be done in a suppository fashion, or as you can see here, as you saw in the previous slide, they can be done orally. These right here are poop pills. They are pills containing fecal transplant matter that can be taken orally in order to replenish the microbiome. Now, unfortunately, in this article, which dates to 2014, they had not yet developed a way to create a capsule that could be colored because the material that the color is made out of needed to exhibit qualities that could withstand stomach acid. And they could not create a colored capsule, but they're working on it so that you don't have to actually see what you're taking when you're swallowing these pills. And I just want to show you guys this cartoon right here, which is kind of a goofy depiction, and it's kind of outdated too because it should be Clostridioides difficile instead of Clostridium difficile. But how C. diff infections can emerge as a result of antibiotic treatment. Okay, that concludes our look at bacterial infections, and now we're moving on to viral infections, of which there are 3. We've got mumps, hepatitis, and viral gastroenteritis, and we'll get started with mumps. Mumps is caused by a virus that colonizes the parotid glands, which are your salivary glands that are just below and in front of your ears. And the fact that it colonizes this area is what leads to the characteristic sign of mumps, which is swelling and pain on one or both sides of the face, because that's where the parotid glands are located that have been infected. So it is transmitted quite easily in saliva and respiratory secretions. It's considered very contagious, and although we associate mumps with this swelling of the face, the mumps virus will disseminate through the body and it can affect other organ systems. For example, in 20 to 40% of men who acquire mumps after going through puberty, testicular inflammation occurs. And as a result of this testicular inflammation, 30 to 50% of those who experience it exhibit at least some sort of testicular atrophy. And sterility can be a rare complication of the mumps virus also. So although mumps is typically mild and is particularly mild when children catch it pre-puberty, if men get it after puberty, it can be dangerous. And so this is one of the reasons why it is included in the MMR vaccine. One of the M's here stands for mumps. It's the measles, mumps, rubella vaccine. Next, we have hepatitis. This pertains to inflammation of the liver. The liver is one of the accessory structures of the digestive tract, and although hepatitis can be caused by chemical toxicity, here we are only concerned with the inflammation of the liver caused by viruses. And there are 5 different viruses that are known to cause hepatitis. They are named hepatitis A through E. So on this slide, we can see a comparison of these different viruses. Hepatitis A is ingested, so it gets into the body orally. The symptoms of Hep A are mostly subclinical, but they include loss of appetite, nausea, diarrhea, fever, malaise, and jaundice, which is a yellowing of the skin. Hepatitis A does not develop into chronic infections, which means that it is cleared from the body at some point. and there is a hepatitis A vaccination that has reduced its incidence greatly. We also have a vaccine for hepatitis B, but hepatitis B differs from hepatitis A in several ways. It's transmitted sexually or parenterally, meaning through breaks in the skin or mucous membranes. The symptoms are similar to hepatitis A, but chronic infection can lead to liver failure or liver cancer because, unlike hepatitis A, Hep B can form into a chronic infection that is not cleared from the body. This is more likely to happen if Hep B is acquired in infancy. 90% of infants infected with Hep B will develop a chronic infection, but only 10% of adults will. Although Hep B is rare in the US due to vaccination, it is still common worldwide, and there are an estimated 350 million chronic carriers of Hep B worldwide. Hepatitis C is transmitted parenterally, so again through breaks in the skin or mucous membranes. It's particularly a danger for people who use intravenous drugs. It is similar in symptoms to hepatitis B, but the thing that is scary about hepatitis C is that it takes an incubation period of 20 years for the appearance of these symptoms and for the recognition of the disease. It also becomes chronic in a large majority of cases. 75 to 80% of cases of Hep C develop into a chronic infection that is never cleared from the body. Hepatitis C, one fact that not a lot of people know about it, is that it kills more people in the United States than HIV and AIDS. There is no vaccine for hepatitis C yet, although this is still something that is being intensely researched. Hepatitis D is transmitted parenterally, and it is particularly dangerous. It's associated with severe liver damage and has a very high mortality rate. It does develop into a chronic infection, but it only infects people who are already infected with hepatitis B. This virus requires a cell that has already been co-infected with Hep B in order to replicate. Hepatitis E, the final form, is like hepatitis A ingested, so it's transmitted orally. It is similar in its symptoms to hepatitis A and is typically mild. It doesn't develop into a chronic infection, but the reason why it is of concern is because it has an especially high mortality rate in pregnant women, over 20%. So I'll give you a moment to take that all in and also reflect on it in this checkpoint here. We don't have a vaccine for hepatitis D despite its very high mortality rate. Why is the development of a hepatitis D vaccine not a priority for the US? Next, we have viral gastroenteritis. This is our third and final viral infection of the digestive system. Viral gastroenteritis is caused by a few different viruses, but the most common ones are rotavirus and norovirus. Rotavirus is associated with a low-grade fever, diarrhea, and vomiting for approximately 1 week. There are approximately 3 million cases annually in the US with less than 100 deaths, so an extremely low mortality rate. It's treatable with rehydration therapy. A vaccine for rotavirus was developed in 2006 and has reduced its incidence by 98%. The reason why this vaccine was a priority is because although rotavirus has a low mortality rate and is treatable with rehydration therapy, it's more dangerous in infants and children and has a higher mortality rate among those groups. Norovirus, this one does not have a vaccine available, and therefore it spreads quite rampantly in the US, particularly during the winter season. It's also known as the winter vomiting bug. It's associated with vomiting or diarrhea that lasts 2 to 3 days. Its incidence is much higher than rotavirus. It affects over 20 million people in the US every year. But again, its mortality rate is extremely low, less than 300 deaths attributed to it. The thing about norovirus, though, is that it is highly persistent on surfaces and resistant to disinfection. And also, Only a few viral particles are needed to initiate an infection. Where some viruses you need to take in hundreds or thousands of particles, the number of infectious agents needed to initiate an infection here is only just a few. And so these are reasons why norovirus has been notoriously responsible for causing outbreaks on the confined areas of cruise ships. This has happened over and over again where cruises have been turned around and quarantined. This was happening with norovirus before coronavirus started doing it. But you can see this article is from 2019. A Royal Caribbean cruise ship ended up sickening 475 passengers and cutting the cruise short as a result of a norovirus outbreak in this confined space where there's a tight community of people who are all exposed to each other. Okay, we've almost made it to the end here. All we have left to talk about are the parasites. We've got the protozoans and the helminths. There are two protozoans we'll focus on. Those are Giardia and amoebic dysentery. Giardiasis is the name for the infection caused by Giardia intestinalis, a protozoan that we've seen before. We've seen that it possesses a special adhesive disc that I'm pointing out here on the slide, which is used to attach to the intestinal wall. And when it touches the intestinal wall, it blocks absorption of nutrients and water. And this leads to diarrhea that can last for weeks, cramps, nausea, weakness, weight loss as a result of lack of nutrient absorption, and one of the signs can be gas or breath that smells like hydrogen sulfide, so it's got that sulfurous smell to it. Giardia is transmitted in cystic form, typically through contaminated water. As we've talked about before, it is the most common gastrointestinal parasite in the United States. And I personally know people as well as have owned animals that have had Giardia from contaminated water here in Arizona. It is typically diagnosed through microscopy of stool samples, and I used to work in a veterinary clinic here in Arizona where it was not uncommon for us to find these little guys swimming around in the stool samples of the animals that came under our care. Our other protozoan disease is amoebic dysentery, caused by the species Entamoeba histolytica. As you can see right here, Entamoeba histolytica is an amoeba that produces pseudopods. It is known for its ability to ingest red blood cells and destroy them, which is part of its mechanism of pathogenicity. It, like Giardia, is transmitted when it is in its protective cystic form through contaminated water or food. And as a matter of fact, a lot of cases of amoebic dysentery are asymptomatic, but symptomatic cases involve severe diarrhea that contains blood and mucus. This amoeba is capable of perforating the abdominal wall and spreading to other organ systems, which can lead the infection to become life-threatening in severe cases. The last, we have helminthic infections, so the parasitic worms. So brace yourselves because these probably will be the nastiest of the bunch. And our first stop here is Taniasis. These are infections of tapeworms usually caused by Taenia saginata, which is the beef tapeworm. It infects cattle, and Taenia solium, which is the pork tapeworm that infects swine. The life cycle of these guys look like this. So a person who is infected with a tapeworm, the eggs will be produced, so they reproduce in humans, and then they release those eggs into the feces, and then those feces will end up in some sort of situation where they are consumed by livestock. The eggs will hatch into larvae, and then they will lodge into the livestock muscles. And then when those livestock are slaughtered and processed for meat, if that meat is not thoroughly cooked, then the larva form can be transmitted to humans. And then those larvae develop into adulthood in humans, where they will then reproduce in the intestines and start the cycle over again. So cooking meat thoroughly is important to make sure that any pathogens and parasites are destroyed. Incidentally, freezing meat for 24 hours also kills tapeworm larvae. So that advertisement or that selling point of saying that meat is fresh and never frozen, that's not really a selling point from the perspective of tapeworm infections. So when it comes to the symptoms of these infections, the symptoms are usually mild if the pathogen remains confined to the digestive tract. But in rare cases, it can migrate out of the digestive tract into other areas of the body. And when it becomes serious is when the brain or the eyes become infected, and that's when the disease can become life-threatening. So in this checkpoint, what I'd like you to do is tell me what are the definitive and intermediate hosts of the pork tapeworm. Next, we have hydatid disease, which is also known as echinococcosis. It's caused by a tiny flatworm called Echinococcus granulosus, and this guy only reaches 3 to 6 millimeters as an adult. So even though it's a flatworm, it is not a long flatworm like the beef and pork tapeworms can be. The life cycle of this disease often involves dogs. It also can be carried by other livestock animals, including sheep, but the eggs are shed in the feces of an infected animal, and then they end up being ingested by livestock or humans. Then the larvae migrate to other parts of the body and form what are called hydatid cysts, which you can see right here are fluid-filled cysts. The symptoms will vary depending upon where these cysts are lodged. In particular, when they form in the brain or the interior of the bones, the disease can become life-threatening. Lastly, we have nematodes or roundworms. And you may remember from our discussion of helminths previously that nematodes are the most common cause of chronic infectious disease in humans worldwide. So we have a variety of nematodes, but we're gonna focus only on just 5 of them. Pinworms are the first one. These ones are caused by the species Enterobius vermicularis, and pinworms are the most common helminthic infection in the United States. The infections are usually mild. What happens is at night, the female pinworm comes out of the anus and lays eggs around the anus, and this causes itching and discomfort. And that itching can then cause the eggs to be lodged underneath the fingernail, and then later it can be retransmitted to the same person or transmitted to another person via the fecal-oral route. This infection affects up to 1/3 of all children in the United States, which is a pretty stark number. And here's a look at what those little pinworms look like. As you can see, they're very small. Hookworm is next. Hookworm is caused by Necator americanus. It is transmitted through the skin via direct contact, usually with soil that has been contaminated with fecal matter. So walking around barefoot on soil that has fecal matter from humans or animals is one common way of contracting hookworm. When it passes through the body, we can see here in this image of a person's foot that they have contracted hookworm. But in the intestines, what it will do is attach to the intestinal wall using special adapted hooks, and then it will proceed to consume blood and tissue in its host. Ascariasis is the name for an infection caused by Ascaris lumbricoides. This is an extremely common helminthic infection around the world. An estimated 800 million to 1 billion people are infected with Ascaris lumbricoides. Typically, the symptoms are mild and may not be recognized until a worm emerges from the body. It consumes digested food in the intestinal tract and usually does not produce major symptoms. So here is a look at Ascaris lumbricoides worms being extracted from an infected person. The last 2 nematodes to talk about are whipworm. Whipworm is caused by Trichuris trichiura. The reservoir of this guy is soil, and it's transmitted through contaminated food. The symptoms range from asymptomatic to cramping and diarrhea, and one rare but serious complication of whipworm infections is rectal prolapse, which is defined as a condition where the rectum becomes detached from the internal structure of your body and then protrudes out of the anus. Here is what a whipworm looks like, and as you can see, they are very small compared to this matchstick here. And then lastly, we have trichinosis caused by Trichinella spiralis. So these guys form cysts that get ingested in undercooked meat, and then they mature in the digestive tract and migrate into muscle tissue where they form cysts. And this is what it looks like. One of those cysts that is inhabited by Trichinella spiralis here. You can see the striation of the muscle tissue around it, and then the cyst is in the middle. And with that last nematode, we have brought ourselves to the end of chapter 24.